Welcome to the Introduction to Physiology PowerPoint. This video is going to cover a little bit of a lot. It covers a little bit of chemistry, it covers a little bit of math, and as we go through this lecture, we're going to cover different aspects of physiology, and it's really going to help set a nice framework to build upon in the coming weeks. So what is physiology exactly? Physiology is really understanding how our bodies maintain homeostasis. What that really means is, how do our bodies stay around a set point? Meaning, how does our temperature stay around 98? How does our heart rate stay around its baseline, whatever that baseline might be for you? And with all of this, we have to understand how our body reacts to external and internal changes in order to maintain that level of homeostasis internally. So the organization that we have in science is the smallest unit, which is the atom, and then it jumps to molecules, cells, and tissues. Tissues group together to create organs, organs come together to create organ systems, and those organ systems create organisms as a whole. In this course, we're going to be looking at cells, tissues, organs, and organ systems. So which systems are we looking into? All of these here. We'll go through each one of these organ systems. We'll talk about their anatomy, and then we'll go ahead and talk about their physiology and how they function, how they communicate with one another, and how they change or alter their functions based on what's happening in their environment. So a basic physiological mechanism is listed here. Let's say you're outside working and it's a really hot day and you're sweating. As you're sweating, you're losing your water from inside of your body through a process called evaporation. So as you sweat, the water comes to your skin. The heat energy causes it to evaporate off. As water is leaving your body as sweat, the fluids and content that stays behind your body becomes more concentrated. In your body, you have receptors that sense the rising concentration in that body, and it sends a signal to the brain to say, hey, our internal environment just became very concentrated. We should stimulate thirst so that we can drink some water. And as that person intakes water, the water intake will help to lower their body concentration. So this whole pathway really is all about maintaining homeostasis. Homeostasis of what? The internal body concentration. This word here is important because it really is what physiology is about, maintaining homeostasis. So what is homeostasis? Well, it's the ability of your body to regulate its internal environment. Whether that's, again, the heart rate, whether that's your respiratory rate or your body temperature, it's your body's ability to make sure that your internal environment is consistent and stable around a baseline, regardless of what's happening in the external world. So, on a cellular level, what does this mean? Well, your cells must also maintain homeostasis relative to their outside world. So the inside world of a cell is called the intracellular fluid, and the outside world of a cell is called the extracellular fluid. This membrane right here, we call it the plasma membrane, also known as the cell membrane, separates the intracellular fluid from the extracellular fluid. Now, why is it so important to separate those fluids? Well, because as you can see here, the extracellular fluid has high concentration of orange and green, and the intracellular fluid has high concentrations of purple. Orange and green are sodium and chloride ions. So outside of the cell, you need to make sure you're maintaining a high sodium and chloride concentration. And inside the cell, you need to make sure that you have low sodium and low chloride concentrations and a high potassium concentration. Please make sure you memorize these concepts. You have to know which ions are high inside and outside the cell. That will greatly help you on your exam. So just to do an overview again, in terms of physiological mechanisms and homeostasis, we need to make sure that we're creating a homeostatic environment. So how does your body do that? Well, your body is constantly getting input signals. So input signals about what your heart rate's doing, about what the concentration of your blood is, about what your body temperature is. All of those input signals end up going to this integration center. And for the most part, in us as humans, our integration center is our brain. So our brain is integrating that input signal and deciding what it wants to do with it. And once it decides, it sends out an output signal, and that output signal is what creates some sort of response in your body. Now, there are two ways in which we can do this type of control. One is called local control, where we don't even go to the brain. We locally understand what's happening, and we can locally make a change and create a response. The reflex control is the type of control that actually sends a signal all the way up to the brain, which is the integrating center here, for evaluation of the input signal. Ultimately, the brain will decide what it wants to do with that input signal and send an output signal to create some sort of response. Now, within those reflex controls, I'm going to go back. The reflex controls we're talking about right now, you have something called a feedback loop. These feedback loops work within reflex controls to regulate how much response is taking place. You either have a negative feedback loop or a positive feedback loop. The negative feedback loop is able to downregulate your output signal, while a positive feedback loop upregulates your output signal. So what's a good just everyday example? So of our negative feedback loop, a really good everyday example is a tropical fish tank. You've got your tropical fish, you've got the water, you've got the thermometer, a wire, a control box, and then the water heater. So tropical fish must live at relatively warmer temperatures of water. Let's say the water temperature goes a little bit lower than what we'd like. Well, that lower temperature is the stimulus. It will be sensed by the thermometer. It will pass that input signal through the wire until it sends that signal to the control box, which in this case is the integrating center. This integrating center will then say, hey, that water temperature is a little bit too low. Let's go ahead and send an output signal, which is going to be turning the heater on in order to create the response of warming the water up. When this happens and this heater turns on, does this heater stay on forever and ever and ever? Well, I hope not, because it would warm up this water too much and the fish would probably die. So there's something called a negative feedback loop. Once this water reaches a high enough temperature, it's sensed by the thermometer again, sending the signal to the control box. And the control box then says, oh, we've reached the temperature we want to be at. We can go ahead and turn off the heater now. This circulating process is called a negative feedback loop. Another example is heart rate or body temperature bouncing around a normal baseline. So let's say you have your heart rate. Your heart rate may drop down a little bit below baseline, then it can jump up a little bit above baseline, and so on and so forth. That is your body's natural way of regulating heart rate. It does this with body temperature as well. Your body temperature and heart rate aren't perfectly exact 24 hours of the day. But what is perfectly exact in a healthy individual is the ability of the body to make sure that we're always going back towards our baseline. So if we're too high, we come back down to baseline. If we're too low, we come back up to baseline. and our example of a positive feedback loop. Positive feedback loops are almost like reinforcing loops. It senses the change and it creates a stronger response every time it senses change. So the example is childbirth. The baby, it'll drop in the uterus. Its head will start pushing on the cervix. When baby's head starts pushing on the cervix, that causes cervical stretch. When the cervix stretches, it will stimulate the brain to release oxytocin. Oxytocin's job is to go back down to the uterus, which is this whole muscle here, and cause contractions. Uterine contractions will then push baby's head further. As baby's head pushes further into the cervix, it'll cause the cervix to stretch even more, which means more oxytocin release, more uterine contractions, and more of the baby's head pushing on the cervix. This process will continue until baby is completely out. Even when baby is completely out, the contractions don't stop immediately. They can continue for a few hours after delivery. So looking back at a little bit of chemistry, we're going to go over just some basics, nothing too in detail. So the first thing that I want to go over are the major macromolecules in chemistry. You've got lipids, which really are our fats. Lipids are made of a glycerol head and a fatty acid tail. And those fatty acid tails look like this. Here's what I want you to know about this. That lipids are made of a glycerol head and a number of fatty acid tails. If you have one fatty acid tail, monoglyceride. 2 fatty acid tails, diglyceride. And 3 fatty acid tails attached to the glycerol is a triglyceride. These fatty acids are hydrophobic. Hydrophobic. We will talk about this word in a few slides. The second macromolecule are carbohydrates, or sugars. Sugars are monosaccharides, disaccharides, that can build up to be polysaccharides. We'll start with our monosaccharides. Monosaccharide just means a single ring sugar. Here are 5 single ring sugars: ribose and deoxyribose, which make up RNA and DNA. Then you have fructose, glucose, and galactose. Now, if you take these monosaccharides and you start bonding them together, you get disaccharides. And di, of course, means 2. A glucose and a fructose makes sucrose, which is our daily table sugar. You do not need to memorize these combinations. Just know that a disaccharide is 2 monosaccharides put together. And then when you build these large structures, we call these polysaccharides. And polysaccharides, dependent upon their structural conformation, will determine what they look like externally. For example, if you're in long, straight chains, you're most likely going to look like something crunchy, like an animal chitin, or you're going to look like glycogen. If you're in loop-de-loop structure, you're most likely going to be a starch. These are all polysaccharides, complex sugars. The third macromolecule are nucleic acids, like DNA and RNA. Nucleic acids are made up of these smaller structures called nucleotides. I'd like you to know the structure of a nucleotide. It is made up of a phosphate group, which is in green, the sugar, which is in pink, and a base, which is in blue. So sugar, phosphate, and the base. You don't need to memorize the chemical structures, just that there is a sugar, a phosphate, and a base. Now, whether your DNA or RNA will determine what kind of sugar you have. If your DNA, it's a deoxyribose. If your RNA, it is a ribose. Those are the 2 kinds of sugars that we have in nucleic acids. Phosphates will always be a phosphate. This doesn't change. And the base has 4 different base options: adenine, guanine, cytosine, and thymine. So I'm going to get my pen here and put a line there. These 4 are the bases that are found in DNA. In RNA, you have adenine, guanine, cytosine, and uracil. So that's for RNA. RNA does not have any thymine bases in it. Instead of any thymines, it has uracils. Now, adenines and guanines fall under the purine group. Why is that? Because purines have 2 rings, and adenines and guanines both have 2 rings. Cytosine, thymines, and uracils fall under pyrimidines. That's because they have a single ring. So all 3 of these structurally have a single ring only. And again, here are your sugar subunits: ribose or deoxyribose. Deoxy means without an oxygen. If you notice, this guy here has 2 hydroxyl groups. This here has only 1. So you've lost a hydroxyl group, and therefore you're deoxyribose now. The last and 4th macromolecule are amino acids. I'd like you to know this structure as well, that the central element is a carbon. It's connected to a hydrogen, an amino group, which is an NH2, and a carboxyl group, which is a COOH. Every amino acid differs in its R group. This is called the variability region. Depending on which R group you have, that will determine which amino acid you are. Now, kind of transitioning a little bit, we're going to go into some basic chemistry: protons, neutrons, electrons, and such. So protons are positively charged, electrons are negatively charged, and neutrons are neutral. Protons and neutrons are found in the nucleus of the atom and really contain all of the atomic mass. This is where we get our mass from in the atom. Why is that? What about the electrons? Well, electrons are super fun. They zip around the outside of the nucleus, and you can have several of them, but they're so tiny and they're moving so fast that we consider their masses negligible. So although electrons do not contribute to the mass of an atom, they really are the single most important subunit in determining how reactive an element is and how that element can bond. If you notice here, you have H2O, an oxygen with 2 hydrogens. The electrons are what hold those elements together. So, what if you have an element and you give it some neutrons or you take away some neutrons from it? For example, you have a hydrogen element. Normally, hydrogen has 1 proton and 1 electron. This is a normal hydrogen from the periodic table. Hydrogens by themselves generally do not have neutrons. But what if I wanted to give it a neutron? I just want to stick a neutron in it. As we know, neutrons do not have charge. So am I changing the charge of my hydrogen? No. But we know that neutrons are found in the nucleus. So what did I change about this hydrogen? Well, I changed the mass. And when you change the number of neutrons that are found in an element, you now call that element an isotope. This is a very important term. Make sure you know the term and the definition. And what if we wanted to change the number of electrons? The number of electrons can be changed as well. You can lose electrons or gain electrons. And did we say that electrons have a large mass? No. In fact, electrons have a very small, negligible mass. So we're not changing the mass of this element. What are we changing when we lose or gain electrons? If you remember, an electron has a negative charge. So if this hydrogen loses an electron, you're losing a negative charge, which means your hydrogen becomes positive. Anytime you gain or lose electrons from an element and change the charge of that element, you are now called an ion. So this here is a hydrogen ion. This here is a hydrogen isotope. The terms ion and isotope are important. Please make sure you know them. Now, as you lose and gain electrons, you lose and gain charge. And both can equally take place. Anytime you have ions that have gained electrons, we call those anions. Anions have gained electrons and therefore they are negatively charged. Cations have lost electrons and therefore they are positively charged. A good way to remember this is if you have lost electrons, you are a positive element. And we think of cats as positive things. They're nice pets to have. So cats are positive. But do you want ants as pets? No. Anytime you have ants in your house, it's kind of negative. It's a negative thing. So cats or cations are positive. Ants or anions are negative. And here are the most common cations and anions we're going to talk about in the class for the semester. So moving on from electrons to types of chemical bonds. These topics kind of relate to each other because it is due to either the sharing or stealing of electrons that you're able to make chemical bonds. So the first kind of chemical bond we're going to talk about is a covalent bond. And the key word here is sharing. This is when two elements are able to share electrons. Now, they can share them perfectly evenly, which means that the electrons flow perfectly evenly throughout the whole molecule. Or electrons can be shared unevenly, like in water, where the oxygen pulls more electrons toward itself and creates a partially negative region and a partially positive region. Anytime electrons are shared perfectly evenly through the entire molecule, that means there's no overall charge because there's no more negative here than there is here. That is called a non-polar molecule. Anytime you have electrons being pulled towards one end of the molecule, creating partial negativity, that is called a polar molecule. The next type of bond is called an ionic bond. Now the key word here is stealing. An ionic bond between two elements usually entails one element stealing electrons from another element. One element outright takes the electrons from the other element, but at the end of the day, both elements are happy. How can this be possible? Well, there is a rule in chemistry that all elements want to have 8 electrons in their outermost shells. 8. It's called the octet rule. So here's sodium, and here's sodium's outermost shell. It only has 1 electron. So sodium either needs to find 7 electrons to gain or lose this 1 electron so we can drop down to this next full energy level. Chloride has 7 electrons in its outermost shell, so it can either kick off the 7 or just gain 1. Sodium and chloride are a perfect match for each other because while sodium wants to give away one electron, chloride only wants to take one electron. So they do the exchange. Sodium gives up its electron to chloride, and as it gives it up, it turns into a positively charged ion because it has given off one electron. And chloride has turned into a negatively charged ion because it has gained one electron. The last two types of chemical bonds are called hydrogen bonds and van der Waals forces. Hydrogen bonds are always in between a hydrogen and, let me draw my lines, F, which is fluorine, an oxygen, or a nitrogen. Anytime there's a bond between a fluorine and a hydrogen, an oxygen and a hydrogen, or a nitrogen and a hydrogen, these are called hydrogen bonds. These are intermolecular forces. So here we have one water molecule. Here we have another water molecule. In between the water molecules, we have a hydrogen bond, bonding the blue hydrogen of one water to the red oxygen of another water. The last one is a van der Waals force. All I really want you to know about these are that these are nonspecific, and they're found in every single molecule you can think of. Every molecule has van der Waals forces. Now, in the beginning of this lecture, I mentioned the word hydrophobic, and I said we'd go over it. And so here's the time when we go over hydrophobic versus hydrophilic interactions. Hydrophobic and hydrophilic can be broken down to really understand the definitions of the words. Hydro means water, phobia is fear, and philia or philic means attracted to. So if you're hydrophobic, you have a fear of water. But if you're hydrophilic, you are attracted to water. So what does that mean in terms of chemistry? Well, let's say you have a beaker full of water molecules. You put in some salt or sugar, let's say. If you take sugar or salt and you put it into a cup of water and you mix, will it eventually dissolve? Yes, it will. Why is that? It is because salt and sugar are both hydrophilic. They both like water. If you are hydrophilic, you are going to happily interact with water and you're going to happily dissolve in water. This means you have a high water solubility. But what if you're hydrophobic? If I took olive oil and I put it into a cup of water and I mixed, is that oil going to eventually mix into the water? Well, no, it's not. It will always separate out. And that's because oil is hydrophobic. If you recall talking about lipids earlier in the lecture, lipids have a glycerol head and fatty acid tails. These fatty acid tails are very hydrophobic. They do not like water. So if you mix them in with a cup of water, they will not dissolve because they have a fear of water. Now moving on to the next topic is going to be acids, bases, and pH. Make sure you know how to use this equation. It will be a math problem on your exam. So acids are any compound that releases H+. These can also be termed protons or hydrogen ions. A base is any compound that accepts protons. So how do I calculate how acidic a solution is based on the number of protons? Well, here's your equation. You're going to calculate what's called pH. pH runs from a scale of 1 to 14. Anything between 7 and 14 is basic in terms of the pH scale. Anything between 1 and 7 is acidic on the pH scale. 7 is our neutral value. So to determine the pH, you need to get on your calculator, and you are allowed to use a scientific calculator, and take the negative log of the proton concentration, which I will give you. So we do have an example problem coming up, so just give us one moment. A basic solution has very few protons, but it can also be stated to have very high number of hydroxide ions. An acidic solution has a lot of hydrogen ions, but not very many hydroxide ions. And a neutral solution has the same number of hydroxides as it does hydrogen ions. Here's a question. What do you get when you combine one hydroxide with a hydrogen? So if you take an OH and you add it to another H, what do you get? You get H2O. That is water. Now, in addition to acids and bases, you have one more factor, and these are called buffers. Buffers can be substances that are able to maintain the pH of a solution. They avoid changes in pH. They help a solution stay at a constant pH level. So a buffer is able to do both, accept protons and donate protons, which is pretty amazing. In our human bodies, the most important buffer we have is bicarbonate. And bicarbonate has the ability to accept a proton to turn into H2CO3 with a neutral charge, or it can lose this proton to become CO3 with a 2- charge. So you see, bicarbonate can take both pathways to help regulate the pH in whatever kind of set point it needs to. Now here is your equation and your example problem. What is the pH of a solution whose hydrogen concentration is 10 to the -7? So pH equals negative log of H plus. Go ahead and plug in 10 to the -7 in your calculator and take the negative log of that number, and it will give you a pH value. The pH of this is going to be 7. I may give you a scientific notation number like this, or just a simple decimal. If you need help with this, please make sure to set up some office hours with me so we can go over it together. And the last topic on this lecture is going to be about proteins and how proteins activate or inactivate. Now, before we can talk about this, we need to understand the function of proteins, and that's going to be to bind ligands. So here is an example protein. Here are some binding sites, and here are the ligands that are able to bind into these binding sites. Now, if you recall from biology, enzymes are very specified types of proteins. So enzymes, their main job is to help reactions move faster by binding the reactants closer together. So how do we get enzymes to either activate or inactivate in our bodies to help chemical reactions move faster, or to not let chemical reactions move faster? Well, there's 3 major ways of activation, and that's going to be proteolytic activation, cofactors or allosteric activators, and then 2 ways of inhibition, and that's going to be competitive or allosteric inhibitors. Let's talk about the activators first. Proteolytic activation. Lytic means to cut something. Let's say this is our inactive protein, and these green fragments, once chopped out, creates this new structure and therefore an active protein. There's something that I need you to write down right now, and it's going to help you for the rest of this class. And it is protein structure dictates protein function. I will say it again. Protein structure dictates protein function. So if you change a protein's structure, as we did here, we change its function from inactive to active. And in this example, with cofactors, here's our inactive protein, and here's the cofactor. The cofactor binds into this groove, and once the cofactor binds, it opens up binding sites for the reactants to bind into. The letter L signifies ligand, also known as ligand. That just means some sort of substrate that binds into a binding site. So because we bound the cofactor to the inactive protein, we changed its structure and therefore its function. We went from inactive to active. And then we have allosteric activators. Allosteric activators are small subunits that bind to the back end of inactive proteins, and when the allosteric activator binds to the inactive protein, it changes its structure almost sequentially, like a domino effect. So the structure changes here, then here, then here, then here, and all the way around. And as that happens, it opens up binding sites. And then these binding sites are there for the ligand to bind. This is called allosteric activation. We went from an inactive to an active protein. Now, two ways to inhibit active proteins is going to be competitive and allosteric inhibition. Competitive inhibitors are substrates that bind to the binding site and block the binding of the actual reactant or ligand. Hence, it's called competitive because it competes with the ligand for that binding site. When the competitive inhibitor binds into the binding site, we've changed the structure and therefore the function of the protein, deeming it inactive. And the last way is called allosteric inhibition, and this is like allosteric activation, but instead of an activator binding to the back end of the protein, you have an inhibitor binding to the back end of the protein. So when this allosteric inhibitor binds to this allosteric site, it's going to change the structure of this active protein into an inactive protein with no binding sites, and therefore the ligand has nowhere to bind. Lastly, with these enzymes or proteins, we have other extraneous ways in which we can activate or inactivate them. And two of the biggest factors are temperature and pH. When temperature is too high or too low, the protein activity will decrease or the protein will break, and we call that a denatured protein. Same with pH. If the pH becomes too acidic or too basic, that protein will denature and therefore not work anymore. And the last piece is saturation. If you have an enzyme, that enzyme can only work to a maximum point. No matter how much more ligand I add to it, meaning reactant I add, the reaction rate is going to go up, up, up until we've reached a maximum rate. That's going to be the rate at saturation. That means at this point here, all of the enzymes are bound by as many ligands as they can bind to, and therefore they've reached their maximum rate of reaction. You've now gone through the Intro to Physiology lecture. If you need any help or have any questions, please don't hesitate to contact me. Thank you.